Hello everyone, welcome once again to the Zoological Society of Assam initiative, a volunteer service to reach out the students during the tough time of COVID-19 outbreak. At the very onset, let me beg apology for the simplicity of the presentation. It was made simple keeping back in the mind the target audience would be undergraduate second semester students as per their syllabus by most of the state universities, and they are just beginners into the zoology major course. In my last presentation, we have discussed about the overview of cell. In case you have missed the description of the video until now, you are unaware of topic of today's presentation, let me throw a quiz through which you can guess what would be the topic. And here is the hint. We are going to discuss today about a cell organelle. Last time we discussed about overview of cell. Today we are moving into a cell organelle. That cell organelle has a link with the DNA in your body. That DNA you received from your mother, not from your father. And if you are a boy, you are not going to contribute that to your children or would-be children. And if you're a girl, yes, you are going to contribute that to your offspring. Well, in a lighter mood, there is nothing to do with the gender discrimination, but because of the process of fertilization. If you can recall the process of fertilization, where only nuclear contribution enters the egg, else remains outside. Well, by the time, if you could guess, yes, I intended to talk about mitochondrial DNA, and yes, the organelle is a mitochondria. However, recent information regarding absorptions of paternal mitochondrial DNA by egg on prolonged attachment have also been reported. But classically, mitochondrial DNA inherited maternally, and we'll go through the presentation with that notion only. So now onwards, in the upcoming 30 minutes or so, we'll look into the general account of mitochondria, little bit history of discovery, general morphology of mitochondria, its membrane and compartment structures, chemical composition of course, mitochondrial DNA as well as mitochondrial ribosome. If you can remember our last presentation and overview of cell, so you can easily figure out the location of mitochondria on cytoplasm by its classical bean-shaped structure with some foldings inside. But with the advent of higher resolution microscopy and modern sample preservation technique, it is found that mitochondria bears highly dynamic structure. In this figure of an electromicrograph of lung macrophage, you can notice bean-shaped and round-shaped mitochondria. Now it is an established fact that switching among different forms of mitochondria is connected with physiological status of the cell and ultimately the organism, and failure of which may lead to disorders like Parkinson's diseases in human. Another aspect of mitochondrial dynamism is that mitochondria interact with other cellular organelles including cytoplasmic reticulum, lysosome and peroxisome through proteins and ions of the organelles. So mitochondria are cytoplasmic energy converting organelles and thus termed as powerhouse of the cell or energy provider. This we'll discuss little later. These are present virtually in all aerobic cells of animals and plants and algae, protozoa and fungi, but absent in bacteria. The numbers and forms of mitochondria per cell depends on the types and functional state of the cell. If you can recall the structure of a sperm, in its neck region of human sperm, the mitochondria are spirally packed. Sperm cells needs energy to swim long passages to reach egg. It is really tough to believe that a few hummingbirds beat their wings 60 times in just 1 second and thus have a metabolic rate 50 times more than that of human. The muscles of wings carry highly packed mitochondria that supply energy to the birds in the form of ATP. Fish gills filament have a high mitochondria-rich cells because it has to deal continuously with the water current for gaseous exchange and maintenance of osmotic balance, and thus in demand of high energy. Eggs of sea urchin contains 140,000 to 150,000 mitochondria, while some algal cells contain only one mitochondrion. So mitochondrial is the singular, mitochondria is a plural term. The shape of mitochondria varies, as already said. It may be filamentous, granular, clubbed, racket, ringed, or vesicular shaped. The size varies in between 0.5 to 2 micrometer, but it may reach up to 7 micrometer as well. Let us have a brief look on the history of discovery. The word mitochondria inherited from Greek mito means thread and chondrion means granule. Kolika in the year 1850 discovered mitochondria in striated muscle cells, but he didn't mention them as a mitochondria. It was a Benda who in the year around 1897-98 stained mitochondria with alizarin and crystal violet. These are some cytological stain, and called them mitochondria. Prior to this cell organelle has been named and renamed with multiple terminologies like phyla, bioblast, vermicules, chondrosomes, plasmosomes, parabasal bodies, and even as fuxinophilic granules. After the discovery of ATP by Carl Lohmann in the year 1929, many path-breaking development occurs in the field of mitochondria, and at late 20th century we came to know about degenerative diseases identified by mitochondria as mentioned by Wallace. The 1997 Nobel in Chemistry was shared by three scientists, Paul, John, and Jens, for their elucidation of the enzymatic mechanism in ATP synthesis and discovery of ion transporting enzymes. Let us have a look on its morphology. Each mitochondria is bounded by two specialized membranes, specially designed for crucial functioning of the cell organelle. The two membranes are smooth outer membrane and the highly convoluted or folded inner mitochondrial membrane. Each of the mitochondrial membrane is about 6 nanometer thickness and of fluid mosaic ultrastructure. The series of inner membrane enfoldings are called cristae. The space between the outer and the inner membrane is called intermembrane space or perimitochondrial space. The inner cristae are towards the space called matrix. Mitochondrial matrix is filled with homogeneous gel-like dense proteinous material. The matrix contains mitochondrial DNA, mitochondrial ribosomes, and granules. So if you zoom a bit of the mitochondrial section, first we would get the outer membrane that contains many copies of purine and enzymes, namely monoamine oxidase, NADH cytochrome C reductase, kynurenine hydroxylase, fatty acid coacetyl A ligase. The next would be the intermembrane or perimitochondrial space containing adenylate kinase, nucleoside diphosphokinase. The inner membrane contains significant enzymes such as nicotinamide adenine dinucleotide, flamin adenine dinucleotide, diphosphoperidine nucleotide dehydrogenase. The DEH here is for dehydrogenase. The short form has been used because of space constraint. Cytochromes BCC1A3, ubiquinone, ATP synthetase, again SYN is for synthetase, succinate dehydrogenase, hydroxybutyrate dehydrogenase, acyl transferase. The matrix contains mitochondrial DNA that we talked initially at the starting of this presentation, with mitochondrial ribosome, tRNA, then malate dehydrogenase, isocitrate dehydrogenase, fumarase, ecotinase, citrase synthetase, ketoacid dehydrogenase, and ions, etc. But why does different kind of enzymes present at different locations of mitochondria? These different structures actually represents the diversity of contents at the mitochondrial compartments. So this is something related to the functions of mitochondria. As this current presentation deals only with its structural aspects, you would be able to understand the function of mitochondria with the subsequent presentations by different teachers in coming days. Just for an idea, an outline of functions has been given, such as outer membrane is associated with mitochondrial lipid synthesis and synthesis of the enzymes that converts lipid substrate. Those are metabolized in the matrix. Intermembrane space deals utilization of ATP molecule passing out of the matrix to phosphorylate other nucleotides. Inner membrane is linked to carry out oxidation reaction of respiratory chain, ATP synthetase enzyme complex that makes ATP in the matrix, transport protein for regulations of metabolites into and out of the matrix. Mitochondrial matrix holds the required enzymes for pyruvate and fatty acid oxidation, citric acid or Krebs cycle. The ATP consists of a purine-based adenine, a pentose sugar ribose, and 3 molecules of phosphoric acid. When the last phosphate group of the ATP is released, large amount of energy comes out and ATP becomes ADP, adenine diphosphate. Well, if you consider mitochondria as a whole, it contains almost 70% protein, 25 to 30% lipid, 0.5% RNA, and a small quantity of DNA with major variations from cell to cell. Outer membrane contains almost half protein and half phospholipids. Porin, multiple copies, is the prime protein of outer membrane, which is a transport protein that forms large channels through lipid bilayer and allows entry of molecules below 10,000 Dalton. The lipid content of mitochondria contains about 90% phospholipids, about 5% cholesterol, and 5% free fatty acids and triglycerides. Cardiolipin is a specialized phospholipid found in inner membrane of mitochondria that makes this membrane impermeable to certain ions and molecules. Let us have a sectional view to peep into the inner membrane and matrix thereafter. Let us zoom a sectional portion. So, by the time you are aware of what is outer membrane, what is inner membrane, and what is intermembrane or perimitochondrial space. Further, the inner membrane has an outer cytosol or C phase towards the intermembrane space. and an inner matrix or M phase towards matrix side. On M phase of inner mitochondrial membrane, at an interval of 10 nanometer, repeated units of stalk particles can be seen. These are called elementary particles or most commonly oxysomes. These are also identified as F0 F1 particle meant for ATP synthesis or phosphorylation and act as ATP synthetase and ATPase. Though not today, but of course it was a surprise in the 1960s that mitochondria have their own DNA known as mitochondrial DNA. Mitochondrial matrix contains copies of mitochondrial DNA which is double-stranded and circular. The picture is a typical vertebrate mitochondrial DNA which is short of about 16 kilobase pair long in human that codes for 37 genes. You can see the location of different genes like cytochrome C oxidase subunit 1, 2, cytochrome B, etc. Similarly, we can get organisms having mitochondrial DNA as small as 2 protein coding genes and as big as yeast having 85 kilobase pair mitochondrial DNA that codes for 30 proteins. In continuation of mitochondrial genetic apparatus, yes, one important characteristic feature is that we mentioned initially, that it is of maternal inheritance. Thus, there is no or rare recombination. And because of this single line lineage, many a time mitochondrial DNA is a good choice for researchers in species differentiation. The 13 mitochondrial gene encoded proteins are responsible for production of protein subunits of the enzyme complexes of the oxidative phosphorylation system, which enables mitochondria to act as the powerhouse of the cell. But mitochondrial genome not able to carry out its functionality only with its self-made proteins. Thus, nuclear gene products are imported into the mitochondria. Generally, one mitochondrion contains many of its genome, and likewise, each cell contains many mitochondria. I have underlined this sentence because it would be needed to understand the two terminologies that I am going to mention in a while. Proofreading mechanism and DNA repairing mechanism is weak in case of mitochondria, resulting to accumulation of mutations. Homoplasmy is when all copies of mitochondrial DNA are same in a cell, be it normal or mutated. On the other hand, heteroplasmy is when copies of mitochondrial DNA are different in a cell, either normal or mutated. In recent developments, it is evident that not only nuclear genome but mitochondrial genome is also linked with many diseases such as muscular weakness, heart disorders, in cases where affected cells utilize more energy than other cells. One well-known mitochondria DNA linked disorder is Leber hereditary optic neuropathy. It's a vision loss associated with homoplasmic mitochondrial DNA mutation, though heteroplasmic transmission also occurs. Interestingly, mitochondria have a process called mitophagy. This is an autophagic pathway through which damaged DNA copies can be removed. In mitochondria, ribosome remains tightly with the inner membrane. Mitochondrial ribosomes, that is mitoribosomes, performs protein synthesis and they have evolved functionally to synthesize membrane proteins. In mammalian cells, mitoribosomes are comparatively small with a total sedimentation coefficient of 55S, composed of two unequal subunits. The small subunit binds messenger RNA, that is mRNA, and translates the encoded message by selecting transfer RNA or tRNA. The large subunit contains the ribosomal catalytic site called PTC or peptidyl transferase center that catalyze the formation of peptide bonds, that is polymerization of the amino acid delivered by tRNA into polypeptide chain. So we are almost at the end of our discussion. So in summary, mitochondria is a specialized cell organelle specially designed for carrying out oxidation, dehydrogenation, oxidative phosphorylation, respiratory chain of the cell. In the process of oxidation of carbohydrates and fats to produce carbon dioxide and water, energy is released, which is utilized by this organelle for synthesis of energy currency known as adenosine triphosphate or ATP. In animal cell, mitochondria produce almost 95% of ATP molecule and thus termed as powerhouse of the cell. In plant cell, in addition to mitochondria, chloroplast also act as transducing system or energy converter, which is absent. Chloroplast is absent in animal cell. This cell organelle is unique, having its own mitochondrial DNA and mechanism for protein synthesis at mitochondrial ribosomes. However, for its entire functions, mitochondria is dependent of nucleus-directed products. It has attracted attention of current day researchers due to its involvement with human pathology. So this is Shamim signing off, giving you a food for thought. Mitochondria are like cell within cell. So this would lead us to our next discussion on semi-autonomy and endosymbiosis of mitochondria. For feedback, feel free to write. Stay safe, take care.